Okay, well, thanks everybody for sticking with us here. We are absolutely delighted and thrilled to welcome our first speaker today. Bryan Grenfell. Grenfell is Professor of Ecology and Evolutionary Biology and Public Affairs at Princeton University in New Jersey. He's a population biologist and studies processes that occur in populations at different scales and how infections move through such groups of organisms. His work is crucial in helping to control disease in humans and animals. His work is theoretical as well as based on large data sets, demonstrating how the density of a population and randomness interact to change the size and composition of populations. And alongside colleagues from the National University of Singapore, he's studied measles in developed countries and is now extending those investigations to whooping cough and other infectious diseases. Amongst other honors, Bryan was awarded the T.H. Huxley Medal from Imperial College London in 1991 and the Scientific Medal of the Zoological Society of London in 1995. And today he's going to be talking about what cross-scale research can tell us about predicting, understanding, and mitigating future pandemics. I couldn't think of a more appropriate talk for our symposium. Bryan, please take it away. Thank you so much.
Okay, so in a short time, I'd like to rush through and talk about the complexity of epidemics, and this audience is very expert on that, and Madhav gave a great introduction yesterday. Sometimes things turn out to be dynamically simpler, and I want to. I'm a traditionalist in this way. I want to advance the idea that we should always compare simple and complex models, and this audience is very apt with that. I know it's sophisticated. I then want to go on and use that to go on and talk about pandemic and epidemic dynamics across scales and how we deal in modeling with particularly immunological and evolutionary complexities. And then I want to spend a chunk of time just on my own thoughts about research and global health priorities, and there'll be some overlap with others, but I'm sure you'll come up with better ideas than I will there. This is, I'm going to touch on work that's covered many decades, a lot of great collaborators and kind funders. So like many people on this Zoom call, we dropped most things in the last year, and this is mainly my colleague Jessica Metcalf and I from Princeton and our groups, and modeled lots of stuff from the local dynamics potentially on the Princeton campus to immunological uncertainties, seasonality, and some quite applied things locally. But the day job really for me for a long time now has been looking at the population dynamics of endemic infectious diseases. And here's my favorite, which is measles. And you see that there appear to be relatively simple patterns here. This is the pre-vaccination era, and you can see that there was an epidemic every other year, more or less, the more annual at the start. This is 1950. Now, can we make simple models of this? You're going to have some temerity on thinking you can, because we know, and Madhav summarized this brilliantly, there's a ton of network dynamics boiling away underneath. There are individual heterogeneities, and measles is a predator of memory cells in the immune system. There's beautiful, intricate immunological dynamics going on, and there are experts at all these scales on the call, which is great. It's great to have that breadth of expertise. I'm gonna try and argue that we can sometimes make simple models, because measles is unusual. This would not be true of COVID-19 or influenza. You don't shed the virus again once you've been naturally infected, and the vaccines are pretty excellent as well. So the immunity is very strong, and it's very, very transmissible. And this is a randomly rewiring network, mainly a childhood infection, and therefore you can sort of close one eye and hope it's mass action at some level, but it's always really important to test that against more intricate models. And a crucial one really is that these epidemics, they didn't do anything particularly strong to control them. They just assumed it was going to happen, and therefore there isn't big behavior change. And behavior change completely complicates things, obviously, and that's certainly been true in non-pharmaceutical interventions in the pandemic. Again, this audience will be very expert on this. Epidemics depend on the reproduction ratio, now in very common parlance, that in an immunizing infection, susceptible and infected recovered tends to deplete susceptibles, therefore the effective ratio goes down when it passes through, when the epidemic drops away. I'm going to persist in calling this the epidemic clockwork, and there's this idea of herd immunity, which you can see strongly in measles and measles epidemics and measles vaccination, for example. So that's the kind of basic dynamical pattern. And again, and then one can obviously make very simple mass action models. And just to show these, it's a very, very simple two-dimensional system, really, with an extra kicker in that there's seasonal transmission, and there's obviously stochasticity. And season, though it's a simple clockwork, potentially, seasonality and stochasticity can introduce lots of dynamic complexities, chaos in particular. So we can take, there are great historical records of measles. Ask me and I can point you at some data we've put on the web recently. But you see that we can make models and run them forward. We can fit them to the short-term behavior state-space models, and they capture this lovely bifurcation from annual to biennial dynamics in this case. And that's because of the baby boom. High birth rate, the epidemics, the susceptibles get topped up quicker, so the dynamics are annual, and then it winds out into this biennial pattern. So this is just a way of saying that there's some emergent simplicity here sometimes under those heuristics that I gave at the start. You might see simple dynamics. So we can use these simple models, and it's really great to have a hierarchy of models where you compare simple and complex models. What's the manifold? Is there a manifold of simpler dynamics in a complex model? And again, you folks are expert on this. This is lovely work by Rachel Baker earlier in the epidemic. There was a lot of discussion of seasonality of COVID. Certainly the endemic coronaviruses are certainly winter diseases. They like low humidity. But Rachel showed nicely, I think, that because you have such violent epidemics, strong susceptible supply at the start, then you wouldn't expect early on, at least, to see the effects of seasonality. In a subsequent paper, later on, and now you do tend to start to see the effects, but the susceptible dynamics would dominate. So that was kind of a use of a simple model. Of course, there are then lots of complexities. The dynamics are kind of okay in the face of these. You can get away with simple models to some extent if you're cautious, but there are super spreaders, age, space, many models for spatial dynamics. The use of cell data records for phones and other things have exploded. They were already revolutionary before the COVID pandemic. They've really exploded then. And non-pharmaceutical intervention, social forcing, max squaring, and so on. I just want to touch on some complexities we've looked at, which make things even more uncertain. We're getting some more data now. The nature of immunity against COVID-19 was not clear. Clinical, was it transmission blocking, pathogen host evolution, and then pathogen community dynamics, which I'll touch on at the end. So this is a lovely paper by Shalisa Roy and Caroline, led by Shalisa Roy and Caroline Wagner, and they made a very simple model where you have you have secondary transmission, and you have the strength of retransmission and the strength of secondary susceptibility, and therefore you can titrate these to go all the way from the simple SIR model to the SIRS model, where you go back to complete susceptibility. And obviously the simple SIR model is more optimistic in terms of the epidemic dynamics. And therefore they were able to look at a weak immunity case where you get recurrent epidemics and a pessimistic case, and a strong immunity case. And the bottom row here is deployment of the vaccine. And you see that there's, again, a much more optimistic case here. In terms of the original strain, the vaccines seem to be doing well against that. So we're in the optimistic case. However, obviously, new strains of the virus are potentially, actually to some extent, but also potentially worrying here. And that brings us to, what are the evolutionary implications of reinfection? In particular, this purple area here, and you can read the paper in Science to look at it, the purple area here are secondary susceptibles. So how important are they in terms of evolutionary dynamics? And this recalls previous work that we've done here, and this is comparing measles and influenza just in a cartoon, both RNA viruses, both imperfect error correction on reproduction. But for measles, the strains, different variants of the virus are different eyes here. But the cross immunity is so strong, you get SIR-like dynamics, and the surface molecule phylogenies, glycoprotein phylogenies, don't have strong patterns. And influenza, because the underlying biological difference is really interesting here. Won't go through it. Because you can get reinfected and retransmit, particularly when there are cluster jumps, for example, you get this lovely ladder-like phylogeny, and you get strong evolution of immune escape. And in this paper, we made a very simple framework where if you have immune pressure, then if immune pressure is very strong, there'll be a lot of strength of selection. And Paul Turner set a brilliant stage for this yesterday. But very low viral abundance, and if viral abundance to some extent controls transmission, you'll select, but you won't transmit. It's the other way around here. There's not much selection and strong transmission, and probably where influenza and maybe COVID-19 are is at some intermediate level where you would have strong transmission of antigenic novelty. So Shadi and Caroline have put this into a paper. This is in medRxiv, and you can look it up. But looking at secondary infectives as the kind of denominator of different sorts. You've had one dose of the vaccine, you've had two, you've had natural infection, and titrating what the threat, and this is just for one city, it would obviously be very spread in different regions, what the threat of potential evolution is. The bottom line of this paper really is, there's obviously a moral case here as well as an epidemiological one, is we need to vaccinate the globe. We need to knock infection down everywhere to minimize new variants. So this figure, I love this sort of figure, and it's been great to see many versions of it on the front page of the symposium and in Paul Turner's talk yesterday. This is for influenza, thinking about the different scales from the molecular level up to the global level and up to the phylogenetic level. And there's a lot of modeling work at the population dynamic scale. There are wonderful folks within host modeling folks who work at this scale. A real gap for me is how we quantify transmission. It's really hard to do, and I think this afternoon's session is going to be very interesting to look at in that sense. So just to bring this all this together, then let me tell you some personal, just idiosyncratic research priorities. Pathogen surveillance is obviously key, and this is really a global health priority. Public health and genomic surveillance, One Health surveillance. Also very valuable for vaccine design, syndromic surveillance. A really interesting cross-scale frontier, and this is a paper by James Hay and Mike Minner and colleagues from Harvard, is a sort of a cross-scale thing where you take the cycle threshold, the CT value from PCR, from pathogen surveillance, and you can read this yourselves. That changes. This is in a model, but they show it in data. That changes over the course of the epidemic. So you can get a feel for where you are in the epidemic trajectory from the CT values as well as from the epidemic pattern. And I think that's extremely exciting. Read the paper. I was very struck by that. That's really the start of a new frontier. Another frontier, I think, is we try to measure epidemic dynamics, often just from the cases. But if you talk to the meteorologists, they measure all the variables, of course, with satellites and other measurements. And with an immunological observatory, and this is again by Mike and championed by him and Jess Metcalf and others of us. If from exhaustive serology around the world, you could measure the susceptible and the infected individuals, you're in a sense not doing an n-step ahead prediction, you're doing a one-step ahead prediction. There's a ton of other good reasons to measure susceptibility and immunity as well. And there are a ton of other things that you want to measure, obviously, in surveillance. Key thing, and this is a very NSF thing, underpinning natural and social science is extremely important. I think as part of the observatory, systems immunology and population level immunity extremely important. And as I've said, transmission biology, that seasonality pattern of the winter virus is lovely work by Jeff Shaman, was just by matching patterns of population epidemics to, and some experimental infections of animals to things like humidity, there's been an explosion now of looking at, for respiratory viruses, at the fluid dynamic aspects, droplet chemistry, all kinds of things, and calibrating that against human movement is a really important frontier, I think. Post-pathogen evolutionary biology, those cross-scale dynamics, very hard to do, but extremely important. I'm not going to touch on it, but social dynamics and how you quantify it, the mask wearing social dynamics, vaccine hesitancy, and so on. Then in terms of models and computation, again, great lessons pioneered in our area, I think, by Jeff Shaman and other, Kat Shea and other folks, Matt Ferrari. Model hierarchies, inference, averaging of models is extremely powerful. Something I always push is we should try and establish dynamical case law when this crisis is gone. We should try and model, as it were, everything. We don't know what the process is and what the threats are. So we should really be modeling all the epidemic phenomena that we see. But we've got to have adequate surveillance to be able to do that. Otherwise, it's really terra incognita. Just to finish off, I talked about polymicrobial interactions, and these are really interesting. A dramatic example of these, there are a lot of dengue and lots of other things. There are lots of polymicrobial interactions. But as you'll probably know, and this is lovely work by Rachel Baker, again, grad student Daniel Park in PNAS, the typical pattern of the very important childhood viral infection, RSV, is the dotted lines here. Everything really in the current year, this is and this is the start of the national emergency of the pandemic. Everything, many other viruses have dropped through the floor. But the trouble is, Rachel runs this forward in the paper. The susceptibles are going to build up, right? And therefore, you predict that you'll get maybe out of normal phase, summer epidemics, for example, RSV is a winter disease. Because people don't get infected then, they'll get infected later on. So there are long-term polymicrobial implications for endemic diseases of these pandemics. And very interestingly now, this is just out, in Australia, they're seeing out of phase southern summer epidemics, which is kind of qualitatively predicted by these models. There's a ton more to do here. So it's a vital and a very exciting area, and the transdisciplinary quantitative work that you folks all do is absolutely key to it. Okay, I'm finished.
Thank you so much, Brian. That was an absolutely fantastic talk. We're going to hold off on questions. We have a kind of combined Q&A session for 20 minutes at the end of all of the talks. So if people want to ask questions, just pop them in the chat there, and we'll be monitoring the questions. We'll collate those. Aditya, shall I introduce Professor Yu, or would you like to do it?
I guess I'll go ahead.
Okay, thank you.
Yeah, thanks, Paul. So I'm delighted to introduce our next speaker, Professor Bin Yu from UC Berkeley. She'll be talking about curating a COVID-19 data repository and forecasting county-level death counts in the US. So Professor Yu is Chancellor's Distinguished Professor and the class of 1936 second chair in the Department of Statistics and of Electrical Engineering and Computer Sciences at UC Berkeley, and a former chair of statistics at Berkeley. She focuses on the practice, algorithm, and theory of statistical machine learning and causal inference. Her group is engaged in interdisciplinary research with scientists from genomics, neuroscience, and precision medicine. She's a member of the US National Academy of Sciences, fellow of the American Academy of Arts and Sciences, and she has won a lot of awards, including being a Guggenheim Fellow, the Tukey Memorial Lecturer. She was the president of the Institute of Mathematical Statistics, and also she received the E.L. Scott Award from the Committee of Presidents of Statistical Societies. So without further ado, I welcome Professor Yu to talk about her insights. Thank you.
Can I share the slide? Yeah. Okay. Thank you very much for inviting me. It's really a pleasure to be here and also learn from all the colleagues. So we are kind of like the non-expert to kind of jump into this COVID prediction and data repository curation last March. Like for 2 months, my group can see that most of my group jumped in with me because we responded to the call by a nonprofit organization. And in the summer, we continued and also got help from Google. We also worked with many other universities. So our motivation was working with this non-profit organization, was very new last March, called Response for Life, to really try to help with the crisis at the time about PPEs. So this is some photo that we work with 2 people from the organization, also Arab Bridge, to ship PPEs for Temple Hospital in May. So here's an overview of what we did. When we joined the organization, there was no data, so we had to curate and get data and use our personal network to get on top of logistics. And then we decided to build at the county level predict, because the organization needed us to help them to prioritize different hospitals so they know where to ship. And we only had data at the county level. So we had subgroup of students just worked on different predictors, transparent and simple, and work on uncertainty measures using prediction residuals, and we run a website. It's kind of an automatic AI system. So here's basically what we curated. It was about 7,000 hospitals, characteristics, and also at the county level, we had many, together about 200 features for the hospitals and maybe 50 for the counties, including socioeconomic factors and many other like mobility. So this is public for people to use. I want to bring up an issue that hasn't come out a lot, which is data quality. That the 2 sources, US Effects, and they're supposed to be a New York Times, supposed to come from the same sources, but they don't agree. And there's also weekend effect, which shouldn't be there. And also people do historical revisions, which basically through our prediction algorithm of, to a very discontinuous, and we make more errors. So these things, I think, is the bottom of all this epidemic prediction is really data quality. I think we need to pay attention to. So as I said, we developed many different predictors. I took advantage of some work I did at Bell Labs 20 years ago for audio prediction, came very handy, which is combined. In the end, we found 2 of them very useful, the linear predictor and the other expanded shared county county level prediction. We just use the neighboring county for a week ago, the data on the deaths and cases, and then the past deaths comes from the same county. So very simplistic. And then we combine depending on which one works better in the past few days, and we weigh them. So very simplistic and kind of engineering and signal processing. To deal with uncertainty, we just also took a very agnostic approach that we just look at the last 5 days, the prediction errors our algorithm has made. And then we took the maximum, and then we add and plus the prediction on top of that. And we can do some calculation under some changeability condition that will give 83% coverage. If you want higher coverage, you can just tune a factor empirically, because the good thing and bad thing about this is that every day you have a new data point, you can kind of tune the coverage. And this philosophy is called conformal prediction without a probabilistic generative model. So you can see that's what we can do. Like this is, you can see the slide was made almost a year ago, but the methodology hasn't changed. And then the paper was written really to support the website and has appeared in Harvard Data Science Review. We're not going to write a paper, but we wrote it because we want our work and the code's reproducible. So we decided to write it up. And this is the website we'll be running called covidseverity.com to support the response for life, but also just to give information. Now there are a lot of good dashboards at the county level. At the time when we were doing it, there was no other county level dashboard. And our predictions are at one of the CDC websites. So we produce our predictions to the CDC website. And if you do a click on the left, you actually can get Arlington County. This is from yesterday. You can see that the blue is the case number, the red is the death, and they're not on the same scale. You have left and right scales. You can see that hopefully things are turning for Arlington County in Virginia. So to take a step back, this is, we constructed an automatic AI system. But every time I give a talk, I go to the website and find something's not quite working, right? So this really says that we need a human-machine collaboration, that you need a human surveillance of the website to correct mistakes. Like, for example, on May 21st, there was an entry of 15,000 deaths from King County in Washington. It's later got corrected to 539. So there was probably a human error, but the machine wouldn't know that and just have a huge jump, and our algorithm just got thrown off. I also want to acknowledge AWS and ISF support, because we're using this AWS computing resource due to a big data grant from ISF, and that's what we write it down. So for our team, we still run this data repository open source on GitHub, and our method very simplistic, transparent, interpretable, and give very comparable short-term prediction performance as agent-based mechanistic model. So I think we should try to leverage both approaches. And we've just finally got access to California hospitalization data, and we're doing some causal investigation. I feel like causality is very hard to establish, but we have to make a decision. So in this sense, it's a causal investigation, because decisions, even you don't do any change, that's a decision, that no policy change. So I think we really need to get evidence to help the policymaker to make informed decisions. Take a step back to look at challenges, opportunities. We really need a nimble national, international surveillance and intervention network system, and has to be complete with manufacturing, supply chain, logistic planning, and stockholder coordination, because we're doing PPE, right? So it was not easy to work with FEMA, for example. Especially without established relationship with them. And the data quality is like the most fundamental first step for all the other systems to be reliable. Otherwise, we might have very propagation of error, human error, as I showed you, and the other errors that are very hard to correct up down the stream. And this has to be in collaboration with the AI system, right? Human is beyond the human scale. So we need a responsible, trustworthy, and reproducible AI system with human in the loop. If I delve into the AI system, I feel like we need a transdisciplinary framework. As already mentioned, we need a coherent and unified technical accessible terminology so the multidisciplinary people can work together and we use the same term in the same thing. And we also need an integrated distributed computing platform for the analytics, or to support the analytics, like data lake, maybe Spark and Databricks. And the approaches should be diverse, agent-based, signal processing, machine learning, and with uncertainty assessment, also risk management capacities. Validation and reality check has to be in the loop and systematically, regularly carried out, and with multiple objectives, health, economy, and fairness. And there's this dynamic change that nobody can predict. So the surveillance and the reaction time has to be fast. When we change things departing from what we know, we have to be agile to adapt for climate change and other possible economic, social, human events. So I'd like to put give a shout out to a framework my group has developed for the whole data science lifecycle. Particularly, you can see what we did is an application of this PCS framework. PCS means predictability, computability, and stability. We're also working on a textbook to really build data science on these basic principles. And a key component of PCS is actually documentation. So I think we also, when we develop this AI system, we need a very good documentation to document all the human judgment calls, as we all know, the modern node we make, and for every step of the process. I also want to bring out the point about humans, right? So if we don't have good human researchers or team members, then we cannot do this. So the culture has to change from winner take all, usually in science, to collaborative culture. And NSF and other funding agencies hope will have visionary and fair incentives, so rewards for grant authorship, reward, and just awards. And I personally think that it's not enough for NSF just to support transformative research, but also have to support verification and replication for trustworthy and reliable research. And we have to really know how the human and machine should collaborate. And education aspect of transdisciplinary research has to be very much at the center for the young generation people to kind of get in and then continue this research. Thank you.
Thanks a lot, Ben, for that nice talk. And I'll give it over to Paul, I think, for introducing our next speaker.
Yeah, thank you. That was an absolutely fantastic talk. And next up, Professor Jordan Pekia is going to be presenting. Professor Pekia is the Thomas E. Golden Jr. Professor of Environmental Engineering at Yale University. His research mixes genetics with engineering to study childhood exposure to bacteria, fungi, and viruses in buildings. Professor Pekia is a member of the Connecticut Academy of Science and Engineering and associate editor for their journal Indoor Air. He's a PhD in environmental engineering from the University of Colorado and is going to be talking about tracking epidemics at the population level through wastewater-based epidemiology. So thank you so very much. Hopefully you're ready to go here.
I am ready to go.
Great, thank you. Welcome.
Thanks. Okay, again, hopefully you can all see me share my screen and you can all hear me okay. I want to thank you all for the kind invitation to speak today about our team's research on wastewater-based epidemiology. So ironically, my day job is sort of to think about pathogens in buildings. And I had some history, though, of thinking about human exposure to pathogens in the sewage sludge. And when the epidemic started, you know, there were two different paths to go, and I had to pick one. And we went down the path of focusing more on wastewater-based epidemiology. So here's an experiment that we did several years ago, and it answers a seemingly obvious question, but a pretty difficult one to get to. That is, what type of microorganisms, what type of viral pathogens specifically exist in wastewater? It's difficult to know because those are difficult to culture, especially with all the different types of strains. We didn't have the genetic tools, capability in databases until just recently. But when we were able to put all that together, deep sequencing, databases and genomes for different microorganisms, we were able to get a bigger list of what types of viruses exist in wastewater. This is interesting because you can think of cities of 1 million people or more, all being contributed into one single wastewater treatment plant that flows into one area. One thing that was interesting to note was the high concentration of coronaviruses, indicating or suggesting at least that they were gut-tropic organisms. And that led into, answering some questions on initial phone calls in March of 2020 about whether this could be detected in wastewater. Now, many groups went through this same sort of evolution, and there are a lot of groups now working on this idea. But the general idea is that you can take effluent concentrations of wastewater as it goes into a treatment plant or some other area as it leaves a building. In our case, we look at sewage sludge. You can determine the concentrations of SARS-CoV-2 in it by PCR, similar to the types of tests that's done for human testing. And when we did that early in March, we found that there was a peak that corresponded to the beginning of the outbreak. And as things calmed down, that peak went down as well. These are just various measurements over here on the left in the red. And then these are measurements of new cases. If we formalize that a little bit more, you can see there's a concordance between the viral RNA and new reported cases. Applying some more rigorous statistical analysis, you can see that early on in the pandemic, we were able to detect these viruses earlier than cases were being enumerated, about a 7-day leading indicator due to the slow nature of detecting cases. Not only can we, is there concordance between this data and new cases, but there's also agreement between the epidemic outbreaks and curves and hospitalizations, at least in shape, with different types of offsets. Hospitalization occurs later as well. This was enough information to put together what we're calling the Yale COVID-19 Wastewater Project. This was funding through the CDC and through the Connecticut Department of Health, with lots of collaborators in School of Public Health, the Connecticut Agricultural Station. And under this serpent premise, I wanted to introduce you to is that wastewater tracks cases, but it's more efficient, accurate, than testing. Now, I'm certainly not saying that we shouldn't do testing. And of course, understanding that an individual has an infection is very important for isolation, et cetera, treatment. But we're all short of data. And I think that in terms of population data, wastewater can make some strong contributions. First, it's inexpensive. Right now, we're monitoring more than 1 million people in Connecticut. It's taking 6 samples per day. Each of these samples cost about as much as it costs to do an individual testing. It can be fast. Testing is still, in many cases, prompted by symptoms, and it takes a while to test that person, to get that data, to schedule testing, to get the data back, and to report that. We can take in wastewater data on one day, and we can report that data to the public later that same day. And in some cases, in some ways, it can be more accurate, certainly, than testing. Surveillance doesn't care whether the symptom is whether it's asymptomatic or not, as long as there's shedding. But it also doesn't care about things that really impact testing and testing volume, like holidays, governments, snowstorms, and all the things that we've been dealing with. There's a relationship between these two. And if you can get to a testing program that is robust and a wastewater testing program that is robust, you can compare the two, and this is just through a simple regression that mostly depends upon lagged concentrations of RNA in the wastewater and some adjustments for the strange testing behavior on the weekends. We get coverages that are above 9.9 in all cases across different wastewater treatment plants for the same model. And some of these treatment plants are order of magnitude differences in sizes. What this can mean immediately, and we, I guess, have to think beyond this pandemic or think beyond this country, is that if we didn't have ways of estimating cases, for example, for New Haven, Connecticut, we could estimate them through our wastewater, and the curve would look like this. The curve actually looks like this. This is the actual cases. This is our estimated cases just from wastewater data. Same thing in Norwich, Connecticut. This is what we estimate the curve to be simply from wastewater data. This is what the real curve looks like. It still captures the same type of behavior. It also is smoothed a bit more because, sometimes these regression analyses are big smoothers. So another practical way that we can do this in the framework that we're working with now in cases and what we have is that it can keep our state up to date. Our state uses cases by data specimen collections. We don't use things that are on New York Times or even that are reported by our state. We look back at the data specimen collection, and that means that, again, there is delays in that test data. So let's take Bridgeport. On February 15th, we had wastewater data for that day and all the days previously. We were able to estimate the cases, and that's what's represented here in these light bars. When you get all the way to the black bars, which is about a week earlier, this is the most updated case by data specimen collection. So it's very useful for us to tell the state, we know that you don't know what happened, this entire week, but we can estimate and roughly tell you, yeah, cases are still going down this week.
Dr. Pesce, you have about 3 minutes left.
Okay, so we look this into policy. We report this work to the Commissioner of Health and the state epidemiology. We talk 2 times a week. We work with CDC. We work with local health directors and hospitals, and we have a front-facing website to the public. You can get there by Yale COVID wastewater. It looks like this. You can click on the towns. You can get the RNA concentrations. You can get the case by data specimen collection. There's all kinds of different ways of looking at it. There are protocols for people to use as well. A few other things that we can do with wastewater data, and this is with Sarah Nason at the Connecticut Ag Station, is we can look at markers of behavior beyond infection. So this is just driving, what traffic looks like. Benzotriazole is an inhibitor that's put in brake discs to inhibit corrosion. That leaks when brakes get used onto roads. Rain washes it off. It gets into wastewater treatment plants. And as an indicator, we can see when the stay-at-home orders stopped or started, the traffic went down. When reopening went up, traffic went up. See the same thing with opioids, and I know many people have looked at this type of data before. Opioid use increased with the outbreak, except for the opioids that were used for elective surgeries. We have early warning systems now, where we're able to look retrospectively back and determine that based on when outbreaks occurred and what the wastewater concentrations look like and how you would model wastewater concentrations in cases together, we can pick a threshold. These thresholds of yellow are 5 cases per 100,000 people. And when cases were low over the summer and when we hope cases are low again over this next summer, we're able to provide 90% confidence to mayors, to health directors to say, yeah, you have just crossed a threshold of 5 cases per 100. We're about 90% confidence that that's something. But we noticed when we did cross these thresholds before, we went from 5 cases to 100,000 to orange, 10 cases per 100,000 to 15 cases per 100,000, and then eventually to more than 100 cases per 100,000. The last thing here I'll tell you about is genetics. And there's a real opportunity here for surveillance of genetics. We have a severe lack of capacity to test, do genetic testing right now for specimens that are collected and for looking at variants. Through the state that I live in, but also throughout the United States. And I just want to show you a comparison. Here's some specimen testing early on in the pandemic, separating the New York clade and the Washington, DC clade. Here's the same thing at the same dates in wastewater, and showing that we could pull genomes out of wastewater. We could separate different types of clades, but there's a lot of problems here. This is coverage, and the coverage is pretty weak. We've solved that problem by just focusing now on the spike proteins, and I think moving forward, we have some good solutions for doing that. So the last thing I'll leave you with is, if I think that we need to move forward, the first thing we need to do is move toward automation, and there's no inherent barrier towards moving to that. Surveillance of other infectious diseases, these are things that don't have testing programs, for sure, is where it can be useful, or in places where SARS-CoV-2 testing is not great. We can track genetic strains and evolutions on a more population level through wastewater. We can even do things like look at gene expression in human cells as marker diseases. Modeling, all the modeling you think about doing with cases and the problems with cases, I think could also be done with wastewater data that doesn't have some of those same issues. And finally, we don't have time to talk about this, but don't get me started on what we could do in the developing world and how important this could be. So here's all the participants. It's been a big collaboration. If you want to find the wastewater website, you can go here. You can put that URL in, you can Google it, or you can go to my website and click at the feet of my dog. Thanks.
Thanks a lot, Jordan, for that really great talk. So Paul, I guess we'll go ahead with questions.
Yeah, and so this is actually going to be a combined Q&A session for everybody that was speaking this morning. So kind of a mix-up of a Q&A session and a panel. So we've had a couple of questions come in through the chat. Krista, one of the questions is from you, actually, if you want to ask it. So one initial question was, if major restrictions will cause a larger breakout of COVID-19 later, how can we predict the magnitude and time of the next breakout wave? And will such restrictions cause more or less total infections? Anybody on the panel that would like to take that one? So how can we predict the magnitude and time of the next breakout wave, and will restrictions cause more or less total infections?
I mean, it depends how the immune dynamics work, and we're only just learning that. The thing that I showed was about another infection that the NPIs against COVID-19 were causing to drop down. I mean, I would have thought that was doable, but it's very interesting, wonderful talk by both my colleagues. It's wonderful to see Jordan's stuff, because again, using wastewater might be a really interesting way to get a handle on that.
Actually, Bryan, while you're talking, there's a question that might be most appropriate for you, which is that recovery from measles meant permanent immunity, and why is that not the case for COVID? So
you've got to be careful with permanent immunity. There's almost no such thing as, for many of these infections, really sterilizing immunity. You see a slight bump in the antibodies if you're exposed, if you've had measles. Slightly mysterious. Measles is incredibly invariant. The surface proteins it has to get into 2 cell types. It's very invariant. It's a systemic infection. Probably isn't one reason for that, but it just seems to be very transmissible, but evolutionarily stuck, which is great news, because if there was loss of immunity in measles, we would have considerable problems, I think. The vaccine isn't quite as good, and there's always people keeping an eye on vaccine immunity waning, but we've not really seen it very strongly yet. But it's a great question.
Yeah, thanks for that. I was actually listening to Science Friday while I was in the car on Friday, and a listener had asked Dr. Fauci, why can't we vaccinate against HIV as well? So I think this has got people thinking about, well, what's the next thing we can vaccinate against, which is really great.
Can I ask Jordan a question?
Yeah,
sure.
While
Go ahead.
I'm talking, it's such a great system. Have you, looking at other viruses or other pathogens? It would be a really interesting time now to look at pathogens which have really been knocked down by the non-pharmaceutical interventions against COVID, and then see if you can get an early warning about the way that they bounce back, particularly out of season. I think that would be a really important thing to do in a very elegant study, since you seem to be well set up for it.
I think that's all a great idea. One limitation that we would have in wastewater is that not all of those pathogens that we care about have gut tropism or enough gut tropism to show up in wastewater. I don't know the reasons, and I haven't thought deeply about it, but we don't seem to see a lot of influenza in wastewater. And we see a lot more coronavirus than rhinovirus traditionally. And my suspicion is that has more to do with celltropism than it does to do with incidence of disease.
Sure.
So if we can figure that out, I think we're going to be in a better footing to extend to different viruses.
And enteroviruses might be interesting, not to hog it. I see you were saying Coxsackie virus a little bit, for example.
That's right. Yep.
Great. Thanks, Bryan and Jordan, for that nice discussion. So I had one question about, and this is for all the speakers, like how much do you have to, you all talked about various surveillance programs, right? So I remember Bryan talking about zero surveillance and like cross-scale surveillance, and Jordan obviously from your wastewater, and Ben also, you had talked about various ways of getting case counts. So how much do you have to deal with concept drifts, right? So this is a term which is popular in data science and machine learning, which is in these surveillance data programs. So like, to give you an example, there have been syndromic surveillance programs for influenza, right? Google Flu Trends, which had to deal with where the relationship of the surveillance data to the disease indicator suddenly changed, right? Due to novel events, maybe the way people were using the vocabulary in the Google Flu Trends case. And maybe here, I don't know if there's a festival or something, or there's a huge influx of people coming to the town. So how do you have to deal, how much do you have to deal with these kinds of sudden novel changes in these syndromic surveillance indicators? Thanks.
I would say that's a really great question, and I don't think that I can get quite maybe to the answer that I think that you're looking for in our case, but partly that's because our experience is we just have tried very hard not to let the concept drift. Because we just, we think that the thing that we started with is fundamentally important, and we just don't have the bandwidth to chase all the other really interesting ideas going on. So that's part of it. But I would say more just, thinking about the data that you get in surveillance. We're an academic lab like everybody else here is, I think, when we find ourselves monitoring, right? More like a commercial lab. And things like supply lines and availability of reagents and how that changes and how trends in doing analysis changes and equipment changes are real. And they really impact that. And it's very hard to push all those continually back as the science evolves and say that we're still monitoring the same way. So I think that's where we've had a lot of difficulty.
There's a nice. Did you want to go ahead?
No, I mean, if anyone else wanted to join, I think Ben wanted to speak or something. Oh,
yeah, I would think that for me, it's always the situation can change that dynamic very fast, but both the concept drift, but just the situation, how people's behavior. The only way we can get ahead is really very agile, like surveillance, and then have the ability to react, right? Basically monitoring and regular quality check on the data and with human in the loop, right? You have to train the human to do these checks. That just like if you look at traditional manufacturer sampling line, there's also drift this and that, right? So they have this quality control procedures in place, probably simpler than what we deal with, but the same philosophy should apply here. It's this regular checking by humans and then discussions of communities. I think John asked an earlier question I saw in the chat box about how do we arrive at coherent, unified, accessible terminology. What I had in mind was more like in work on precision medicine, they actually have this category or dictionary of medical terms, some committee agreed on, right? And there's different, for medical claims, you have this classification. So the community, we have to get together, just have a document and be reviewed periodically also by experts. And that's the way I think people can refer to and go for young people come in. That's the way I think we can move, make a difference. It's a committee and basically define some dictionary terms.
Thanks for responding to that question. I wonder if I could ask a further question of Dr. Pekia, a very fascinating talk. I'm guessing that the RNA that you get is extracted from virus particles, because I'm imagining it would degrade if it were just naked RNA in the wastewater. Does anyone look at the infectious activity of those virus particles at all? Because virologists commonly are aware of only a small fraction of total particles being infectious. So there's something called a particle to frequency ratio. Does anyone ever make this kind of measurement for the particles from wastewater?
For SARS-CoV-2, that's been done pretty rarely. Even if you go back into the more obvious case and say just looking at infectivity in human stool, you know, for a long time the dogma was there's lots of virus RNA and maybe viral particles, but nothing is infective. I'm not infectious. I'm not sure that that's true. It's awfully difficult to test for infectivity in stool in wastewater.
Yeah.
It's my understanding, and I think Krista follows this field pretty closely as well. She may know that there are some cases where where there's infectious SARS-CoV-2 in human stool, but nobody's found it in wastewater, and nobody particularly is looking because I don't think it's a high value target for the people who have the capability of doing this.
Great, thanks.
So, Paul, did you want to ask a question?
Yeah, I did actually, because it's an socio-behavioral and economic sciences question. So it's actually for Bryan, but I think Professor Yu might be able to also chime in, because I saw some GIS visualizations on your really amazing dashboards. So the question is, how do we integrate social and behavioral sciences into these models? At an institutional level, how do we get quantitative social scientists to engage social dynamics, behavior change, etc., fundamentally change transmission dynamics and complicate control? This seems like it should be a priority, but we don't see a lot of progress. And maybe if you could just talk about what do you need from the socio-behavioral sciences? What could those disciplines be providing in model form or in quantitative signatures that would help you do more with your work?
So let me try to, so we did collect some, public socioeconomic factors, right? And then we put in our model, it just didn't help. So I think it has to be a more sophisticated way of understanding the dynamic. We also put in some mobility. In the end, it seemed to be all got embedded into the neighbor county's case and death number, right? That's what gave us the best predictive result. But that's only for prediction. But if you want to go for interpretation, then there is policy change. So that's what we have some follow-up project. You have a little bit, not completely quasi-experiment going on there, because the counties make different decisions and change their policies. So I think we have to formulate a team. Maybe we should talk afterwards. Is how do we establish some causality, like evidence that replace a lot of this neighborhood counts, that's what we use, into something more interpretable. And we tried the machine learning way, this didn't help the prediction. So we went for the simpler model. But if you do this traditional statistical testing, many of these demographic and socioeconomic factors would turn out to be, significant, which I don't exactly trust. So we just decided to go for prediction. For the purpose, it worked. We support the PPE distribution, but not mechanistic. So I think we have to integrate some of these models in a more spatially coherent, maybe a Bayesian hierarchical model way. We just did very much signal processing way, one county at a time, because we had the time constraint of I have to do things in a few weeks.
Just to expand on that, I think that's a great answer. My great colleague at Princeton, Simon Levin, is very interested in this. And we have a great student, Virgin Yang, who's looking at the dynamics of mask wearing. And there's a massive data about different countries and different trajectories of mask wearing. And what are the determinants there in terms of social norms and government actions and so on. I get the impression the pandemic, like with many things, it's a really sweet time to think about this now, because a lot of social scientists, a ton of really great economists, for example, are interested in analyzing those sorts of data. I mean, if I had a question for them, it would be, how does a particular intervention, for example, modulate transmission? Are people risk averse? Are they risk seeking? And even qualitative answers like that would be kind of useful. So I think it's an exciting time.
Yeah, I think there's many ways you could do it as well. One way would just be to build an agent-based model of absolutely everything that would have a one-to-one mapping to reality. I mean, we're possibly getting there, you know, in the future, maybe in some areas. I know your ex-colleague at Princeton, Yanis Kavrakidis, had done a lot of really great work on equation-free modeling. So the idea that you can pick up signals of some of these other systems and project them to a manifold, and then from that manifold you can get into other models, and that's quite promising. Yep. Had worked with him and some civil and mechanical engineers on trying to do that for agent-based modeling. There's lots of ways that we could possibly do it. We have 132 participants on this workshop here, so I think together we can probably kickstart something that could help some promising research along these avenues.
I think what you bring up, Paul, is really how do we integrate the agent-based models with the machine learning signal processing approach. I mean, with the time we had, we couldn't really get into the literature to know when you have to work with a group who already have an agent-based model. Yeah. What my thoughts are is that we can do very well follow the trends to do a short-term prediction. And can we kind of use that to constrain the agent-based models beyond just choosing the social network or the other parameters you tune? So use it more as a constraint that to combine that way.
I wanted to point out, so I noted before, I've spent quite a bit of time in research thinking about microbes in buildings, and not always very specifically about viruses and things like influenza or common cold or rhinovirus. But, you know, for many, many years, everybody has known the tremendous human health toll from respiratory infectious diseases. And, you know, part of my tradition comes from environmental engineering where, you know, we would never tolerate that type of disease level in drinking water, but we do tolerate that in the indoor environment for some reason or another. I am hopeful that, you know, one of the silver linings that could come out of this pandemic is that we no longer tolerate that anymore. And in order to do that, we're going to have to break some of the rules that have, you know, and a way of doing things that we've done for so many years. And people in the natural sciences are just going to have to find a way to pull in people from the social sciences or allow them to come in, or whatever the barrier has been. You know, we're never going to get there with respiratory disease unless we have a big contribution from the social sciences and behavioral sciences.
Can I just add to that? I mean, I agree, though you have to be really perfect then, right? Because the susceptibles are going to build up. And if it gets reintroduced from an animal species, you're in trouble again in some circumstances. So I agree with you, but let's have vaccines for everything in as much as we can as well.
Yeah.
So that we've got multiple ways of controlling things.
I think this has been an excellent panel, and I wanted to just take a moment just to acknowledge once again and to thank all of our speakers for the remarks that you provided, for helping to inform our conversation, to spark new ideas and new ways of thinking, and also for taking the time to be responsive to all of the questions that came in from our webinar participants. So thank you all very much again. We're hoping that all of you will be able to continue to remain with us a little while longer to participate in the breakout sessions. We know that we'd love to continue to benefit from your perspective. So thank you once again.